Okay, in this video we're going to go into more detail about the process of actually making urine. So where does it start? How does it become what we're all familiar with? There are three basic processes involved in making urine: glomerular filtration, tubular reabsorption, and tubular secretion. Now, I have these listed as step 1, step 2, and step 3, and calling glomerular filtration step 1 is really appropriate because the fluid that becomes urine has to come from somewhere, and it comes from plasma, cell-free and protein-free plasma, that is pushed out of the glomerulus into that capsular space. It's called filtrate at that point. Listing tubular reabsorption and tubular secretion as step 2 and 3 is really a little bit less reflective of what's actually happening. It's not like tubular reabsorption happens first and then tubular secretion happens. Really, reabsorption and secretion are kind of happening continuously along the nephron. And to remind you, reabsorption is the process where the kidneys reclaim substances that your body wants to hold on to. And secretion is the process by which the urine is fine-tuned according to the needs of the body. So if you need to get rid of excess ions, they can be gotten rid of through secretion. If you need to get rid of drugs or waste, they can get eliminated through tubular secretion. So I want to stress the point that filtrate is not the same as urine. Urine comes from filtrate, but there's modifications. So you could really almost look at it as urine, what you actually eliminate from your body. That basically is filtrate minus the things that have been reabsorbed, plus the substances that have been secreted. This image gives you a look at where these different processes happen. Filtration happens at the renal corpuscle, where you have the glomerulus, it's kind of hidden inside there, and the glomerular capsule. In the proximal convoluted tubule, you have reabsorption. Actually, most of the reabsorption happens here. Water, various ions, and organic nutrients are taken back up. In the descending limb of the loop of Henle, you have reabsorption of water. In the ascending limb of the loop of Henle, you have reabsorption of solutes, including sodium, salt, sodium chloride. In the distal convoluted tubule, you have reabsorption, water, sodium, calcium, and you have secretion. This is also where, if you look here, the arrows that have this kind of dotted line, that indicates variable reabsorption. So where you have reabsorption that varies according to hormonal triggers and according to the needs of the body. Well, I should say according to the needs of the body, which sometimes trigger release of hormones, and that adjusts the secretion or reabsorption at the kidney. For example, parathyroid hormone. Parathyroid hormone, it's released when your calcium levels are too low, and parathyroid hormone tells your nephrons to reabsorb more calcium. That's an example of hormone-dependent variable ion reabsorption. Now over here in the collecting system, including the collecting duct and the papillary duct, you have water reabsorption, ion secretion and reabsorption. So these arrows are moving both ways. Reabsorption is taking solutes or water out of the urine and bringing them back into the body. Secretion, taking solutes from the interstitial fluid and secreting them back into the urine. The water reabsorption that occurs at the collecting duct is variable, and it's dependent on antidiuretic hormone. Water reabsorption up here in the proximal convoluted tubule is constant, so there's always water reabsorption up here. The quantity of water reabsorption in the collecting duct is variable. Now, I want to take each of these processes, each of these three processes, and talk about them in turn. A little bit more detail on what's going on, a little bit more detail on what regulates them, as well as what structures, what aspects of the kidney support these processes. So that means that the first process we're going to look at is glomerular filtration, the legitimate first step in producing urine. Glomerular filtration is a passive process. Your cells don't need to expend any energy, they don't need to expend any ATP in order to produce filtrate. Okay? So keep that in mind. Passive, by passive I mean no energy expended by the cells doing this process. When we talk about tubular reabsorption and secretion, we will be talking about some active processes where energy is expended in order to carry out that step in urine formation. Glomerular filtration depends on the filtration membrane. Remember, the filtration membrane is the barrier to filtration. And a couple of characteristics of the filtration membrane that make the glomeruli very efficient at producing filtrate is that there's a very large surface area, so basically there's lots of holes, lots of filtration slits and fenestrations for plasma to pass through. The other thing that helps out is that there's a large surface area, and that surface area is extremely permeable. So the fact that there are those fenestrations, those pores in the capillary endothelium, the fact that there are those slits between the foot processes of the podocytes, highly permeable. Apart from the characteristics of the filtration membrane, glomerular filtration really, really depends on the high blood pressure in the glomerulus. And this high blood pressure means that there's a high hydrostatic pressure, which is basically just a high fluid pressure that forces fluid out of the fenestrations and filtration slits. The glomerulus is a high-pressure capillary bed, and what makes a high-pressure capillary bed is a difference in diameter between the afferent arteriole and the efferent arteriole. So you have a large diameter afferent arteriole, and then you have your glomerulus, and a relatively small diameter efferent arteriole. So you have a lot of blood coming into the glomerulus and just less blood being able to leave the glomerulus. So that blood flow gets backed up a bit in the glomerular capillaries, raising the blood pressure in those capillaries. Now, if you think back to the cardiovascular system and learning about bulk fluid flow at capillary beds, you can start to understand the interaction of different forces at the glomerulus, the forces that can affect glomerular filtration. So you have this hydrostatic pressure, that's these dark blue arrows. This hydrostatic pressure is basically pushing fluid out of the glomerulus. And there are two forces that oppose this hydrostatic pressure, two forces that tend to keep fluid in the glomerular capillaries. The first is hydrostatic pressure in the capsular space. There is a low amount of capsular hydrostatic pressure, significantly lower than the hydrostatic pressure of the glomerulus itself. There's also the colloid osmotic pressure coming from proteins, primarily from proteins that are present in the glomerulus, in the glomerular capillaries. Proteins, remember, if you recall, proteins can't cross the filtration membrane. So they stay in the blood and they exert an osmotic pressure that tends to draw fluid back. So there's this balance, this balancing between the hydrostatic pressure in the glomerulus, the osmotic pressure in the glomerulus, and the hydrostatic pressure in the capsular space. And when you add these together, you take the blood hydrostatic pressure, you subtract the colloid osmotic pressure, and you subtract the capsular hydrostatic pressure, you get what's called the net filtration pressure that works out to, with these numbers, about 10 millimeters mercury. So the net filtration pressure, the balance between these three forces, does force fluid out of the glomerular capillaries into the capsular space, where it then passes on into the proximal convoluted tubule. Net filtration pressure influences the rate at which filtrate is formed, the glomerular filtration rate, which in turn, the glomerular filtration rate influences the effectiveness of tubular reabsorption because it controls how quickly urine, or filtrate in the process of becoming urine, how quickly that's passing through the convoluted tubules and loop of Henle in the nephron. Glomerular filtration rate is the volume of filtrate formed each minute in both kidneys. And typically it's about, there's variation, but it's about 125 milliliters a minute, or about 180 liters of filtrate formed each day. Now consider that you only have approximately 40 liters of fluid in your body, total fluid in your body at any given time. I think we can pretty much agree that it's a good thing that the vast majority of water in that filtrate is reabsorbed. So the glomerular filtration rate depends on the net filtration pressure, as I mentioned. If filtration pressure goes up, and remember I mean that balance between blood pressure in the glomerulus and osmotic pressure and capsular hydrostatic pressure. If net filtration pressure goes up, the glomerular filtration rate goes up. And the primary determinant of net filtration pressure is glomerular blood pressure. Glomerular filtration rate can also be affected by the permeability of the filtration membrane. So if the filtration membrane is damaged, say, and becomes more permeable, then the glomerular filtration rate will go up. It can also be affected by the total surface area of the filtration membrane. If you increase the surface area, you increase the glomerular filtration rate. But the primary regulator, the primary way that glomerular filtration rate is regulated is through adjustments to the glomerular blood pressure. And it's important that the glomerular filtration rate be tightly regulated. If the GFR is too high, the urine or the filtrate is passing through the nephron too quickly to reabsorb what your body needs to keep. So if the filtration rate is too high, you'll be losing nutrients and ions and water before you even get a chance to reclaim them. On the other hand, if the filtration rate is too low, everything's going to get reclaimed. Everything. Urine will pass through the nephron so slowly that there's plenty of time to take back up everything out of that urine. Even the waste molecules that your body needs to get rid of. One tool that your kidneys have that they can use to control the glomerular filtration rate is the Juxtaglomerular Complex, which is also sometimes called the Juxtaglomerular apparatus. I apologize if I slow down as I'm saying juxtaglomerular. It's a bit of a tongue twister. The Juxtaglomerular Complex, or JGC, consists of the juxtaglomerular cells, also called granular cells, the Macula Densa, and the extraglomerular mesangial cells. So let's take a quick look at where those are located. Now, in previous diagrams of the nephron, we've shown the nephron all stretched out in a nice linear fashion, but the nephron actually turns back in on itself, so that the arteriole leading into the glomerulus is nestled right up against the distal convoluted tubule. So you still have urine flowing through the nephron like that, but the nephron itself doubles back on itself so that the glomerulus, the glomerular capsule, and the distal convoluted tubule are right nestled up against each other. And at this point where they come together, that's where you see the juxtaglomerular apparatus, right in that little space between the beginning of the distal convoluted tubule. And sometimes it's actually referred to as the ascending loop of Henle. Remember that the nephron is a continuous tube. So it's a bit arbitrary to say, like, okay, now we're in the loop of Henle, now we're in the distal convoluted tubule. Okay, so some textbooks will say beginning of the distal convoluted tubule, some textbooks will say end of the ascending limb of the loop of Henle. The point is, this later part of the nephron comes into close association with the afferent arteriole leading into the glomerulus. So you have the granular cells, or juxtaglomerular cells, which are actually part of the wall of the afferent arteriole. They're smooth muscle cells. They're a little bit enlarged and modified in the wall of the afferent arteriole. You have the Macula Densa cells, which are in the tubule, the beginning of the distal convoluted tubule, or end of the loop of Henle. These are the Macula Densa cells. And in between, you have extraglomerular mesangial cells that sit in between the two. You also have, these are referred to as extraglomerular mesangial cells because you also have intraglomerular mesangial cells, or just mesangial cells, that are sitting in between the capillaries of the glomerulus. These mesangial cells within the glomerulus adjust the surface area of the capillaries. They can constrict and basically change the diameter of the capillaries, changing the surface area. And by changing the surface area of the capillaries, you're changing the surface area of the filtration membrane, which can affect the glomerular filtration rate. So these guys have some role in affecting the GFR, but what I really want to focus on are these ones. So let's go back and take a look at what they do. So the juxtaglomerular cells they contain mechanoreceptors. And what they do is they detect blood pressure in the afferent arteriole. And remember that the main determinant of net filtration pressure is blood pressure in the glomerulus, and the blood pressure in the afferent arteriole is going to affect blood pressure in the glomerulus. So these granular cells, they can detect changes in that blood pressure. And what they do when they detect, when they're stimulated, they produce a molecule called Rhiannon, which activates another molecule called angiotensin. And we'll come back to this renin-angiotensin pathway. It's very, very critical in maintaining blood volume and maintaining blood pressure. The other component of the juxtaglomerular complex is the macula densa. So the macula densa contain chemoreceptors that detect the sodium chloride content of filtrate. So basically they detect the solute concentration or the osmolarity of the filtrate. And that's a surrogate for basically how concentrated that filtrate is. And remember, since these are in the distal convoluted tubule, this is after a lot of reabsorption. This is nearing the end of reabsorption. So the macula densa are basically evaluating, they're basically determining how much solute, how much sodium chloride has been reabsorbed out of that filtrate. So you can think of it as the granular cells, the juxtaglomerular cells, are basically monitoring net filtration pressure, And the macula densa, those cells are monitoring reabsorption. The extraglomerular mesangial cells are thought to mainly help by passing signals between these two cells, by allowing them to communicate with each other. So the controls that adjust the glomerular filtration rate can be divided into intrinsic controls and extrinsic controls. So the intrinsic controls primarily act to maintain a steady glomerular filtration rate. So the intrinsic controls, these guys, they preserve kidney function. That's what they're all about. Making sure that that glomerular filtration rate does not get too high or too low. The extrinsic controls, in contrast, these are all about regulating systemic blood pressure. Okay, the extrinsic GFR controls, they're not primarily concerned about kidney function. They've got the bigger picture in mind. They may control systemic blood pressure, they may maintain systemic blood pressure, even at the expense of kidney function. And they primarily do this by decreasing fluid loss. You know, if you've just been stabbed and you've lost a ton of blood and oxygen isn't getting anywhere in your body, you've got bigger concerns than, is my urine too concentrated? Dehydration is not your primary worry at that point. What you need to do is make sure that your circulatory system can keep functioning. And the extrinsic GFR controls adjust what's going on in the kidney in order to help out that maintenance of systemic blood pressure. Okay, so let's go back and talk about the intrinsic GFR controls for a moment. Maintain a steady glomerular filtration rate within a pretty tight window, and these intrinsic GFR controls work quite well within a range of arterial pressures. Then about 80 to 180 millimeters mercury, the intrinsic GFR controls can keep a steady glomerular filtration rate. And these intrinsic GFR controls, they include myogenic mechanisms. Remember, the prefix myo means muscle. So these are mechanisms that are just reflexive actions of the smooth muscle cells around the afferent arteriole. Basically, if blood pressure goes up, then pressure in the afferent arteriole goes up, and that triggers a reflexive constriction of the afferent arterioles. Now, you may be thinking, well, that's weird. Why are you constricting? Isn't that just going to raise blood pressure in the arteriole? And yes, but what it does is it forces some of that blood to go elsewhere, to keep moving it so that it doesn't enter the glomerulus. Right? So this constriction of the afferent arteriole reduces blood flow to the glomerulus. And since blood flow goes down, that leads to a decrease in the net filtration pressure. Okay? Conversely, the flip side of that coin, when blood pressure decreases, that would tend to bring the net filtration pressure down, so glomerular filtration rate would slow down. And this causes a reflexive dilation of the afferent arterioles, which is going to increase blood flow and will correct glomerular filtration rate by bringing that net filtration pressure back up. So those are your myogenic mechanisms, just reflexive actions of the smooth muscle cells. And these act within normal parameters of blood pressure fluctuation, like if you're exercising versus relaxing in front of the TV. Myogenic mechanisms help control your glomerular filtration rate within that range. The tubuloglomerular feedback mechanisms are the mechanisms that involve the JGA, the juxtaglomerular apparatus. Notice the term tubulo, because you have the tubule involved in glomerular, you have the glomerulus involved. That's a key indicator that you're talking about the juxtaglomerular apparatus at the junction between the glomerulus and the distal tubule. So this mechanism is triggered by the Macula Densa cells. And remember I said the Macula Densa cells basically monitor reabsorption through the proximal convoluted tubule and the loop of Henle. And they do this by detecting sodium chloride concentration. Sodium chloride concentration varies with the glomerular filtration rate. If you have a high GFR, fluid is moving through the tubules quickly, so you have decreased reabsorption, which means that you're going to have greater concentrations of sodium chloride in the filtrate, right? The Macula Densa cells detect this elevated sodium chloride, which, remember, acts as an indicator of an elevated glomerular filtration rate. And so when they detect high sodium chloride levels, they release a molecule that triggers vasoconstriction. It's thought that that molecule might be ATP. So the afferent arteriole constricts, blood flow decreases, blood flow to the glomerulus decreases, and you get decreased net filtration pressure, which of course leads to a decrease GFR, bringing the GFR back to where it needs to be. Now for the extrinsic GFR controls, working to maintain systemic blood pressure. We have the sympathetic nervous system and we have the renin-angiotensin pathway. So the sympathetic nervous system, remember that's your fight or flight response. It's stimulated by extreme stress or emergency. It's also stimulated by acute blood loss. So acute blood loss stimulates your sympathetic responses. Key hormones involved in the sympathetic nervous system are norepinephrine and epinephrine. And in the kidneys, these two hormones cause a strong constriction of the afferent arterioles. So this decreases filtrate formation. So the reason why this is helpful in maintaining your systemic blood pressure is because if you have decreased filtrate formation, you have less water being lost to filtrate. That water, that fluid, the plasma just stays in circulation and can continue back out of the kidneys around to the organs that need blood. Now, the sympathetic nervous system also has an effect on the renin-angiotensin system. And the renin-angiotensin system is our second extrinsic GFR control. This again involves the JGA, because remember the granular cells of the JGA produce renin. And these guys detect low blood pressure. It actually, low blood pressure decreases stretch of the granular cells, which triggers the granular cells to release renin. Now there's actually 3 triggers for renin release. The granular cells, this low blood pressure detected by the granular cells is one. The Macula Densa cells detecting a low sodium chloride concentration is another. Remember, a low sodium chloride concentration means that there is too much sodium chloride reabsorption. Actually, I should reverse the direction. Low sodium chloride concentration comes from a high sodium chloride reabsorption, which comes from a low glomerular filtration rate. So that low sodium chloride concentration in the distal convoluted tubule indicates a low glomerular filtration rate. So the granular cells, the macula densa cells, and sympathetic nerves can all trigger the release of renin. But when you have all of these activated, you get the strongest release of renin. Now let's move to the next slide and talk a little bit about what renin actually does. Renin ultimately activates a precursor molecule. So renin takes this precursor, angiotensinogen, and converts it to angiotensin 1. Okay? And angiotensin 1 is converted in the lungs to angiotensin 2. And it's converted by this enzyme, angiotensin converting enzyme, most creative name ever, which is actually found in the lungs, in the endothelium, capillary endothelium of the lungs. So, once angiotensin 1 is converted to angiotensin 2 has this whole set of effects, and all of these effects together are geared towards increasing blood volume and thereby increasing or restoring blood pressure. Okay? So you have these effects of angiotensin 2. You have systemic vasoconstriction. Vasoconstriction raises the arterial blood pressure. You have stimulation of sodium reabsorption, and when you get sodium being reabsorbed, when you get more sodium reabsorption, water follows sodium back. This is, of course, osmosis. And that sodium reabsorption is effected in a couple of different ways. So angiotensin 1 acts directly on the renal tubules to increase sodium reabsorption. It also stimulates aldosterone, and aldosterone increases sodium reabsorption. Angiotensin 2 also acts on the hypothalamus in a couple of different ways. It activates the release of antidiuretic hormone so that I shouldn't say it activates the release, because you always have some antidiuretic hormone. What it does is it increases the release of antidiuretic hormone so that you are retaining more water from your urine. It also activates the thirst center so that you are stimulated to bring more water into your body. Renin-angiotensin system, angiotensin 2 also acts on the kidneys. So it constricts the efferent arterioles, and this constricting the efferent arterioles actually decreases the pressure in the paratubular capillaries so that you get more fluid moving into the paratubular capillaries. It also causes contraction of the glomerular mesangial cells, decreasing filtration surface area so that you get a decreased glomerular filtration rate and less fluid lost to filtrate. Okay, so we've talked about glomerular filtration. We've talked about all of the factors that go into the glomerular filtration rate. Net filtration pressure, the adjustments in net filtration pressure, the intrinsic and extrinsic GFR controls. Now I want to talk a little bit about tubular reabsorption. So tubular reabsorption is taking substances out of the filtrate back into the circulation. Substances can be moved through the epithelial cells of the nephron. Most substances move through the epithelial cells. That's called the transcellular route. So that's this one right there. Actually, that's not a very good color to use, is it? There we go. A few substances, some ions in water, are reabsorbed through the paracellular route, which basically means between epithelial cells. So that's this route. I do want to remind you of a little bit of terminology with regards to the epithelial cells. Make sure you keep these straight as you're studying reabsorption. So the face of the epithelial cell that is in contact with the filtrate, that is facing into the lumen of the tubule, is referred to as the luminal membrane. The other side of the epithelial cell, the side that faces the interstitial fluid, where the paratubular capillaries are, is called the basolateral membrane. Now, normally, under normal conditions, all glucose, all amino acids are reabsorbed. a significant amount, almost all of the water is reabsorbed, although there's some regulation, so there's some variability. And ion reabsorption, there's a lot of ion reabsorption, which can also be regulated, can also be variable. Most reabsorption occurs in the proximal convoluted tubules, and reabsorption begins as soon as filtrate enters the proximal tubule. It's worth noting that wastes are actually reabsorbed as well, but it's not a complete reabsorption. So under normal healthy conditions, urea, for example, you only reabsorb about 50%, about half of the urea that's in your filtrate. Now, reabsorption is basically driven by active transport of sodium. This is an ATP-dependent process. That's what I mean by saying active transport, is that your epithelial cells of your proximal convoluted tubule actually burn some ATP in order to reabsorb sodium and then set up gradients that drive reabsorption of other substances as well. So, sodium active transport sets up a number of different things. That sets up a chemical gradient. A chemical gradient drives secondary active transport. So, as sodium moves down its concentration gradient, it pulls other substances along with it. Right? That secondary active transport is how glucose and amino acids and some ions and vitamins get taken up across the luminal membrane, so into the epithelial cell, and then they can be transported out the basolateral membrane into the interstitial fluid. Sodium active transport also sets up an osmotic gradient that is necessary for the absorption of water. So as sodium is pumped out into the interstitial fluid, that increases the osmotic the osmolarity, the solute concentration in the interstitial fluid, and that pulls water out into the interstitial fluid. Lastly, it sets up an electrical gradient that pulls some anions, some negatively charged ions, and mostly we mean chloride, out of the filtrate and into the interstitial fluid. I should mention that the sodium active transport happens at the basolateral membrane. So if we've got our epithelial cell, that's a microvilli, because we're talking about the proximal convoluted tubule. Sodium active transport pumps sodium out of the basolateral membrane into the interstitial fluid. Now let's take a little bit more visual look at these gradients that are set up. So here we can actually kind of see what's going on. You've got the lumen of the tubule over here with its filtrate. You have the epithelial cells here. You have the interstitial fluid and the paratubular capillary here. These I've circled to indicate where the gradients that we talked about on the previous slide are. So this A here corresponds to the chemical gradient that brings things over with secondary active transport. B is the osmotic gradient, and C, this is the electrical gradient that pulls negatively charged ions into the interstitial fluid, right? And it all begins here with the active transport of sodium on the basolateral membrane. This slide also contains a step-by-step walkthrough of reabsorption in the proximal convoluted tubule. Now, in case you're not quite following how active transport of sodium in the basolateral membrane, how that affects transport at the luminal membrane, I just want to map out the concentration gradients. So, as sodium is being pumped into the interstitial fluid, what you're basically doing is you are setting up a concentration gradient of sodium. So, So as the concentration of sodium is increasing over here in the interstitial fluid, that concentration of sodium is decreasing inside the epithelial cell. Okay? And so, since the concentration of sodium is going down inside the epithelial cell, you're actually setting up a second concentration gradient at the luminal membrane, where there is now less sodium inside the cell than there is in the filtrate. So sodium will diffuse into the epithelial cell and be and pull along with it and co-transport. The sort of the force of the sodium diffusing down its concentration gradient brings these other substances along with them into the epithelial cell. Once these substances are in the epithelial cell, they can be transported across the basolateral membrane into the interstitial fluid through various mechanisms depending on the substance, active transport, facilitated diffusion, exocytosis even. As far as the osmotic gradient that draws water across the epithelium back into the interstitial fluid, as you're pumping sodium into the interstitial fluid, you have an increase in solute concentration. That increased solute concentration will draw water across the basolateral membrane through aquaporins, through water channels, into the interstitial fluid. As water is drawn out of the epithelial cell, the fluid in the epithelial cell becomes more concentrated, and you get water being drawn through osmosis again into the epithelial cell. This third gradient, the electrical gradient that draws anions into the interstitial fluid, is easily understood by the fact that sodium is a positively charged ion, a cation. So you have an increase in positive ions in the interstitial fluid as sodium is pumped out, and that draws negative ions, negatively charged ions, into the interstitial fluid. This is all perfectly fine as long as you have a healthy individual. However, one thing that you should note about that previous slide is that most substances that are reabsorbed require transport proteins. Right? So this is a physical process. It's not magical. You need transport proteins to transport substances across the plasma membrane, substances like glucose and water-soluble vitamins and amino acids. You need transport proteins to carry these guys across the luminal membrane. You even have transport proteins for water called aquaporins. Now, if you have a limited number of transport proteins in the plasma membrane, and all of those transport proteins are occupied, you can't absorb any more of that substance, no matter how much of that substance is in the filtrate. Okay? So under normal healthy conditions, there's as many transport proteins as you need. Under normal healthy conditions, you've got enough, you're fine. But when you have pathological conditions where you have higher quantities, higher concentrations of a particular substance in your filtrate than you would normally have, transport proteins can be exceeded. Transport proteins can be saturated, fully bound up by their substance. And if that's the case, you've reached maximal reabsorption. And that's the excess is just going to be excreted in your urine. This is why in diabetic patients with super high levels of glucose in their blood, that means they have super high levels of glucose in their filtrate, and they have maxed out their ability to reabsorb glucose. That's why they get glucose in their urine. This is also why if you take massive quantities of a water-soluble vitamin, like vitamin C, there are transport proteins that can pick up vitamin C and reabsorb it. However, if you've taken massive quantities of vitamin C, you're going to have massive quantities of vitamin C in your filtrate, and you won't be able to reabsorb it all, and you will have lots of vitamin C in your urine. This idea of a transport maximum and needing transport proteins for reabsorption is also key for understanding how hormones can alter reabsorption. So you may reach the transport maximum and have lots of a substance staying in the urine, but if you add a hormone that increases the availability of transport proteins, so you have a lot more transport proteins in that luminal surface, well, now you can get more of that substance being reabsorbed and less excreted in the urine. This is what happens with parathyroid hormone. Parathyroid hormone increases the expression of calcium transport proteins. Okay. Aldosterone increases the expression of aquaporins, those water channels, so that you can get more reabsorption of water. All right. This slide and the next couple of slides talk about where reabsorption happens along the nephron. It really happens in a variety of locations, but the type of reabsorption that happens varies a little bit. As I've mentioned, the proximal convoluted tubule is where most reabsorption happens, so you get a lot of nutrient reabsorption. You also get reabsorption of a lot of the water and sodium, reabsorption of bicarbonate, and reabsorption of other ions, chloride and potassium. You also, actually at this point, this is where you get some waste products being reabsorbed, uric acid and urea, even though much of that waste is later secreted, back into the filtrates. In the loop of Henle, you have differences in reabsorption depending on whether you're talking about the descending limb or the ascending limb. In the descending limb, you have reabsorption of water. In the ascending limb, you have reabsorption of solutes, primarily salts, sodium chloride. And in the next video, which talks about urine concentration, we'll talk more about how these differences in permeability set up a increasing osmotic gradient as through the interstitial fluid. In the distal convoluted tubule and the collecting duct, you have variable reabsorption, and this is in response to various hormones. So aldosterone affects the uptake of sodium. It also affects the secretion of potassium because it uses these sodium and potassium ATPase pumps. So they basically exchange sodium and potassium. So the reabsorption of sodium is coupled to the secretion of potassium. Increased reabsorption of sodium means increased secretion of potassium. Antidiuretic hormone also acts on the collecting ducts, increasing water reabsorption. Parathyroid hormone increases the expression of calcium transport channels in the luminal membrane of the distal convoluted tubule so that you get increased calcium reabsorption. Okay, so we've talked about glomerular filtration exhaustively. We've talked about tubular reabsorption. I just want to touch briefly on tubular secretion. Tubular secretion is basically the reverse of tubular reabsorption. It happens largely at the proximal convoluted tubules and some secretion at the collecting ducts too. And there are a variety of different substances that are secreted this way. So some protein-bound substances can be gotten rid of through secretion. Some reabsorbed waste, urea and uric acid primarily, are eliminated by secretion. So you get a lot of the urea and uric acid reabsorbed in the proximal convoluted tubule and then secreted later. Seems a little bit wonky why your kidneys would do this, but that reabsorption and resecretion of urea and uric acid actually allows your kidneys to control the osmolarity that helps with reabsorption of water. Excess potassium is gotten rid of, as I mentioned, with aldosterone. when blood potassium levels get too high, aldosterone increases potassium secretion in exchange for increased sodium reabsorption. This is also important for controlling blood pH because you get active secretion of hydrogen ions when your blood gets too acidic. Alright, there's a lot of material in this video. That's because the physiology of the kidney is very, very vital for life, and it's pretty complex. Use your objectives, break it down into pieces, and then build those pieces back together again to tell the story of glomerular filtration, to tell the story of tubular reabsorption and tubular secretion. As far as specific questions that you should be able to answer from studying this video, you should be able to answer these questions about glomerular filtration and the filtration rate. You should be able to discuss the control of glomerular filtration. And you should be able to also explain the role of tubular reabsorption, tubular secretion, and how active transport of sodium affects tubular reabsorption, how that kind of drives tubular reabsorption. And bringing it back to the clinical relevance, tell me why diabetics have sweet urine.